Hey everyone, so today we're going to be talking about the lab equipment used for today's experiments. And as a refresher, this is the Nanomedicine and Biotherapeutics lab. So today we're going to be going over some of the basic equipment that's being used for cell culture, as we'll be going over later in the section, how to pass cells, how to count, as well as how to know if your cell line is healthy or not. So we're first going to start off with the incubator. Hey everyone, so we're going to go over the incubator. The incubators are ideal ways to keep cells under control. And what I mean by that is cells need 37 degrees as well as 5% CO2, so humidity, to grow very well. Those are the ideal conditions, as well as that allows for the carbonic acid buffer solution to maintain ideal pH. And so when we open this up, you'll see that there are some cells in here currently growing. So whenever you're going into this incubator, it's very important that you're spraying your hands with 70% ethanol, because this allows you to get rid of any sort of contamination that you may be bringing into the incubator, such as bacteria, viruses, or whatever. And you have to remember that this is a common shared equipment. So this means that everyone will be using this, so this would be like multiple people within a lab. So you want to make sure you're treating this with care, and that you're being, courteous to other lab mates. And so before I go in, spray my hands, make sure I get ethanol all around. And so I'm going to open this, but I'm more importantly opening this to show you guys that there's this water tray. So this water tray, usually you want to fill it like a third to a half full of water. This will be very deionized millicube water, because you don't want any sort of water that's going to contaminate and get into the cells. And so this is what helps establish the humidity for the cells that you'll be growing later in this quarter. So primary HeLa cells, as well as NK92. And so here are just the two different types of cell culture techniques people could hold their cells in, so TFLAS or wells or tissue culture plates. But once you're done with this, you always want to make sure you're closing this up, and that you'll close this door. And overall, this is what a lot of different labs, even besides ours, and as well as pharmaceutical companies, will be using to help establish their cell lines. So we have here a desktop centrifuge, and so basically what this will allow us to do is look at 1.5 and 0.6 milliliter microcentrifuge tubes. The bigger centrifuge that we'll be showing later in this video, those are actually for 15 and 50 mil conical tubes. And so as you can see on this machine, the left knob will control the time, and the right knob will control the speed at which the tubes will be spinning. And they're going through centripetal force, which will allow the density to separate between liquid and solid. So when we're separating our cells, the cells will actually be located on the bottom of the tubes, while the supernatant, which is the medium, which will be located near the top. And one thing to keep in mind is when you're doing this, RPM is not the same thing as RCF. So when you're spinning your cells, you need to make sure you're very careful about what speed this is on, because if you spin them too fast, your cells will lyse and die. And so for this, we hit the blue button over here, which is the open. This pops the top off, and then you can see here that this top for the microcentrifuge tubes is tightly secured. You always want to make sure that this is taken off first, and then that you'll check and make sure your tubes are balanced. So as you can see here, we're taking off the top, and this will allow us to put our microcentrifuge tubes into the centrifuge. And you'll want to make sure that they're balanced. If they aren't balanced, the centrifuge will start to make very weird noises, and sometimes the tubes can break. And once you have all the tubes balanced, I like to go 1 and 13, and then make sure that they're mirrors of each other. You'll tighten the lid back on. And then you'll close the incubator and start your run. Once this is finished, it will beep and let you know that your samples are ready. So let's start to talk about centrifuges. Centrifuges are important because these are one method that we can do to isolate cells and pellet them down. Centrifuges are important because they spin with centripetal force, so it allows us to separate everything by density. So cells will always be denser than liquid. So those will go to the bottom. And then when we're getting rid of our cells, which we'll be talking about in a different video, we'll kind of go into more detail about like how we want to practice aseptic technique. And so currently when you look at this centrifuge, it has a red button. That just means that it's currently off or sleeping. So when you hit this, green, it comes on. So with our centrifuge, there are different programs which you can save, but that's not really important. Overall, we have 3 different speeds: RCF, rad, or RPM. So whenever you're using this, you always want to make sure that you're looking to see what speed it's already set at. Because in most situations, and when you're in a lab, there will be multiple people using your centrifuges. So it's really important to ensure that, like, if you're splitting cells and you want to spin them down, they're hopefully either at 500G or 1200 RPM, because someone maybe could be spinning down vesicles, and those could be done at like 3000G for all you know. And if you spin your cells too fast, those will lyse and burst and your experiment will be ruined. So it's always really important to like take the time to look at this. And then on our third part over here, this just is the minutes/seconds. So I have it set for 5 minutes. And if you'd like to change this to RPM, you quickly just hit the speed button and that will allow you to change to that. And if you want to change back, you'll just change and then you'll do start. And then so start and stop. So whenever you're using a centrifuge, you always want to make sure that you're around it, because you never know if something is unbalanced correctly. And so when you do put your samples in, wait till everything comes to top speed. If you don't see it like vibrating or making weird noises, you can know that you set everything in correctly. Now looking into the top, you always want to make sure you're opening this carefully because it is very heavy, and also you don't want to damage the machine. So as you guys can see inside the centrifuge, there are 4 different types of, there's 4 buckets here. Buckets are just things that we use to hold our conical tubes or epidors. And so these are made for 50 mil tubes, and then these are made for 15 mil tubes. So it allows us to spin like higher volumes if we're trying to separate our cells or any other solutions that are present. There are also adapters that allow you to spin down 96 well plates, as well as filter plates or anything that you need. For the smaller centrifuges, those allow us to spin down microcentrifuge tubes. And so overall, this is just really important, and you want to make sure you're taking care of it. When you are complete and everything is done, you slowly just want to let this close, and then you know you're good to go. Hey everyone, so we're going to quickly go over some of the lab equipment that will be used in experiment 1. So a lot of this equipment that we will be explaining is not only important for lab 2, but also the other labs throughout the quarter. So we just want to kind of give you guys a brief introduction about, what the equipment does and why it's important and how it will help us to visualize our samples. So here we have the inverted microscope. This is actually a very useful equipment because it allows us to visualize morphology of cells, as well as characterize some of our other cell structures that we may see throughout the quarter. And so here we're starting off with our lenses. So you always want to make sure that you start with the chem wipe and that you'll wipe the lenses of any sort of dust or debris that could be present. And this will just help you like visualize the cells as well. Also with the light switch, so there's an on and off switch located on the side of the microscope. And so this knob controls the brightness intensity. So currently you won't be able to see any light at all, but the minute I dial this knob, you can see that the intensity of the light increases significantly. So this is sometimes what people will do to help visualize what's on the plane of their cells or what they focused on. And so now I want to show you guys some of the microscope, the stage, and below. So I need you to come to the other side so we can better focus on showing this there. So here we have the stage. This is what we'll be using to kind of hold our cell culture plates. And then underneath the stage, you guys will be able to see the objective lenses. So there are 4 objective lenses that range from 4x all the way to 40x. So these just allow us to like zoom in and out of our samples, so you'll be able to see like cells much more clearer. And also here are the fine tune and adjustment knobs. So again, besides changing the objective lens here, You'll also be able to go in and out of focus of your sample, so this is actually very helpful to know. And then the fine-tune one is like if you're almost in focus but not entirely. And also, like, whenever you're dealing with the microscope, you want to be very careful because, you know, these are very expensive and there is glass. So, you know, treat other people's equipment very responsibly. And also, when you are keeping samples on here, you can spill, so just make sure to wipe up anything with ethanol. And then once you're done, make sure to turn off the power switch. And then also, don't forget to put this on top because it's going to prevent any dust from accumulating into the lenses. And this will always be done at the end of the class sections. Hey everyone, so here we have the Biosafety Cabinet. This is primarily what you'll be using for passing your cells as well as many of your experiments. The reason why we want to use this Biosafety Cabinet is because it not only protects the cells from bacteria and viruses, but also allows for a clean workspace. And so, just briefly to kind of talk to you guys about this before I open the hood, there are HEPA filters located at the top of the Biosafety Cabinets. And what's nice about it is you can have recirculation of air and not have to worry about any sort of pathogens, as I said earlier. And so what happens is air from the system, so the surrounding background, will come into these front vents, the grills right here, will go in, and as well as some of the recirculated air from HEPA filters, as well as working in the system, will go into the vents up to the back, and it will go through the HEPA filters to then get purified and allow us to have a clean space. Something you want to keep in mind when you are working in this hood, sudden movements will disrupt the airflow, so you want to make sure you're very precise about what you're doing. And you don't want to constantly like wave your hands around because, as I said before, that will mess up the airflow. And so in order to open this sash, we're going to look up, we're going to raise it to this notch over here. You'll start noticing it as I open, but what's nice about it is it gives us the indicated level at which it's safe to operate. And as I open the hood, you'll start to hear an alarm. That's letting you know that the machine's turning on. And as I said before, now the machine is on, and what's nice is before working in it, you need to wait till these smart flow indicators turn green. That's what indicates that it's safe to work in this space and that the airflow is established. If it is not established, you do not want to be working in the Biosafety Cabinet until it's completed. That we have established our safe airflow as indicated by here, all the dots are green. That means we can now start putting things into the hood. So, as with any situation, you always want to make sure that anything you bring into the hood is sterile. So we're going to spray everything we bring in with 70% ethanol. Not only that, but also our hands, because we're working with cells and we don't want to contaminate them at all. So, for example, I'm bringing in this Eppendorf, which is going to allow me to use serological pipettes. And also some serological pipettes, which is going to help me in terms of passing the cells later today.